Раз, раз.
Hello, hello. It's good. Is there anybody from Latin America here? No. You don't have Latin Americans. We have to change that.
We can start. Everybody can hear me. Yeah, everybody. It's working.
Hello.
Hello, everybody. It's my pleasure to introduce Professor Daniel Rivera.
Do I have a laser pointer?
Daniel Rivera is working in Havana University in Cuba. and short biography. Uh, he graduated from University of Havana in 2002 with first honors, and then he joined a group of Professor Wes John in Germany in Leibniz Institute, where he completed his PhD. Then he performed his independent career back on Cuba in Havana University. He was appointed the president of Cuban Chemical Society. He is the youngest president of Cuban Chemical Society, and Daniel is a renowned specialist in multi-component reactions and
I could say so, yeah.
microcyclizations performed with multi-component reactions. I suppose many of you in this room somehow are involved in development of new multi-component reactions. So I hope you will find some of your works may be suitable for microcyclizations too. Dear students, please try to think about this in this way. We are not having only students, and people are also watching us from the online translation. So,
but we
the
don't
word
have
to
a
Professor
point
Rivera.
analyzer, yeah? I should have. Okay. No, because sometimes I want to highlight something. Okay, good afternoon, everybody. It's my pleasure to stay here, and I want to thank the Dean for the invitation to present our work, what we do in Cuba, but also in cooperation with the Leibniz Institute of Biochemistry in Halle in Germany, where I am mostly a group leader there, directing research, dealing with infection diseases and so on, so on. So today I'm going to talk about chemistry, of course, but I'm going to talk about also the biological applications that we focus in our group, and mostly about vaccine development, okay? Most of us, when we think about a vaccine, we always think that it's a purely biological product. I will show you today how chemistry is very, very important for vaccine development, especially for the type of vaccines that we are highlighting, okay? As was mentioned, thank you by the introduction, by the way, I am coming from this small island in the middle of the Caribbean Sea. I am full professor of the University of Havana. It's an old university for the standards of America, of course. There is a new continent and so on, okay? But I also work, as mentioned here, it's an institute near Berlin at the Department of Bioorganic Chemistry. I am the director of one global health center. It's called for training Latin American students to develop new generation vaccines. And this is important because what I'm going to talk to you is related to that. It's a project that covers the whole Latin American continent to train people from Central America and from other areas to know how to learn vaccines. Perhaps you know that Cuba, besides being a very small country, a very poor country, I have to say, with a lot of very bad economical situation, is very famous because of the political crisis, a typical crisis with the United States, the embargo, the blockade, everything you know very well. So most of the results that I'm going to present today are derived from the fact that Cuba has been forced to develop its own vaccines because you cannot purchase vaccines from American companies. And this is a situation in which at some point you can live without potatoes or without milk, but you cannot live without medicines. So Cuba for 50 years was forced, either you develop your own medicines or then you don't have medicine because you cannot purchase from American companies. This is a situation that led us to develop ourselves, our own technology, and biotechnology development of Cuba is inspired or is derived from the situation that we were forced to do it. And nowadays, Cuba, I'm pleased to say, I will finish that in my presentation. we developed our own COVID-19 vaccine, for example, and that was the result of 20, 30 years of development in the biotechnology field. And I will talk to you today how chemistry contributes to that in Cuba. Okay, the next one. So I will give 3 courses. On Wednesday, I will talk about multi-component reactions in the development of peptide pharmaceuticals. I am mostly a peptide and protein chemistry, but I also work with the small molecules, heterocycles, and so on. So that I will give an overview about peptide macrocyclization, peptide pharmaceutical, peptide drug conjugates, how we develop antibody conjugates to anti-cancer peptides for directed anti-cancer therapy, and so on, so on. That will be on Wednesday. And then on Thursday, I will talk about metal catalysis, and we will talk about how we develop, we and others, this is a more general lection, but very deep to the students about chemoselective methods for the late stage derivatization of peptides. It's like how we can actually derivatize peptides to make it more metabolically stable, more bioactive using transition metal catalysis. But today, we are going to talk about the development of antibacterial and antiviral vaccines, okay? Next one, please. So first of all, because I asked you what was the biological background here, I am also a chemist, who by force we had to study immunology and biology. We have to define by simplicity what is a vaccine, okay? And we have to see that it's a biological product that is used to induce an immune response that confers protection either against the infection or the disease. Remember that nowadays, for example, the COVID situation is that it doesn't matter if we are vaccinated, we will get infected, but probably we will not have a very strong disease, okay? Vaccines sometimes protect against infection and sometimes only against the very severe disease, okay? And the vaccination, we have to see to understand what is the vaccination process, we have to see that this is a training, it's a process of training our immune system. So we have to train our immune system to be in contact with something that looks like a pathogen for a second encounter of our immune system with that pathogen to be able to respond very fast and with high efficacy against the disease produced by that pathogen, okay? That's why we call it, by simplicity, that is a training process. That's why we use not only 1 dose of a vaccine, sometimes 2, sometimes after 10 years we have to give a booster, what is called a vaccine booster, to keep training the immune system on updating the antibody response level, how our antibody elicit, our body elicit antibody response against that pathogen, okay? But a very important part of the vaccination process is what is called the immune memory. Our body has to be able to remember the pathogen, and this is the key for all vaccinations, okay? Today we are talking about vaccine for children, and we will see how we want to vaccinate children of 1 year to make that children immune against the disease for the rest of the year. And all the chemistry I'm going to talk about today is directed to favor the immune memory, to allow our body to recognize the antigen of that vaccine and to remember for the rest of the life, which is the purpose of a vaccine, okay? This is the definition of an antigen. This is very basic for the terms of vaccinology, but for those that are not recognized, so a pathogen sometimes, an antigen sometimes is only a part of the pathogen. It does not have to be the entire virus or the entire bacteria. Sometimes it's a part of the bacteria and that is used by a human to induce an immune protection. The next one, please. So this is a, can we switch off the lights? No? Not possible? So this is a representation of the 2 different types of vaccines that are recognized in the world. This is what is called the live attenuated vaccines. And these vaccines, now you see better? Yeah. So the live attenuated vaccines are those that contain the entire pathogen in an attenuated form. So sometimes you modify some genes of the pathogen to make it non-replicated or to make it less pathogenic, and then you inject, okay? Remember that the vaccines were developed, the concept of vaccination was developed by the English Dr. Edward Jenner as far as in 1798, and was developed for the smallpox disease. That is the only infection disease that has been eradicated by vaccination in humans, okay? But there are also what we call the inactivated vaccines. They also contain the whole pathogen, the whole organism, but in an inactivated form. And these inactivated forms can be by heat, but also can be by chemical treatment, for example, with formaldehyde. Then you kill the pathogen and you inject. You are injecting the pathogen, but the pathogen is not able to colonize your body or to self-replicate inside our cells, okay? But these are what we call the old vaccines. They are still active. Our children still get it, but the next generation vaccine, the next one, are this one that I'm introducing to you. Next one, please. So the third generation vaccines is when you take only a part of the pathogen. For example, a protein that surrounds the virus. This was very common in the COVID-19 vaccines, that we know that the spike protein of the virus is this protein that surrounds the virus, was the antigen that was selected for eliciting an immune response against the virus and was able to protect, okay? Sometimes you isolate the protein by, for example, this is the case of the diphtheria and the tetanus vaccines, in which you grow the bacteria and then you isolate these proteins and you detoxify the proteins and you inject it in our children. All of us are vaccinated against tetanus and diphtheria in that way. These vaccines have more than 80 years of development and they work very well. Sometimes you can express the vaccine, the protein, by recombinant means. I'm not going to talk about that because we are going to talk about chemistry today, okay? Sometimes you can produce subunit vaccines, virus-like particles, outer membrane vesicles. The outer membrane vesicles is a type of vaccine in which you isolate the antigen. The antigen is anchored to the membrane of the bacteria, and then you use that for the vaccination, okay? But the next generation vaccines, next one, please, are those that are called the 4th, the 5th, and the 6th generation, are the protein polysaccharide conjugates. As you see here, we have a covalent linkage between a protein and a polysaccharide, so there is a lot of chemistry behind that, and we will talk about that. We have the viral vector vaccines. This was developed for Ebola, but these are the type of vaccine that Russia also developed, the Sputnik vaccine, but also the AstraZeneca vaccines, and a few of them, the nucleic acids and the antigens, okay? We are going to talk mostly about the conjugation technology, how we chemists contribute to the conjugation chemistry to the development of vaccine, but I wanted to introduce the different families of vaccines for you to see in which the chemistry actually plays a crucial role for the development. The next one. Okay, so this is, as we are going to talk about bacteria, let me introduce you these 3 bacteria. Okay, these are 3 bacteria that kill more than 20 million children per year in the world, okay? Mostly not in Europe. In Russia, you don't suffer from them, but when you take a look to Africa, to Latin America, and to Asia, you see a lot of this. This is Neisseria meningitidis, Haemophilus influenza, and Streptococcus pneumoniae. These 3 bacteria collectively, they are massive children killers, killer of children, okay? They kill more than 20. And the key point of them, they produce meningitis, pneumonia, sepsis, bacteremia, what we call that the children really gets a meningitis that kills 10% of the children that get it. And the key of this is that these bacteria colonize our nasopharyngeal tract. So when we kiss our children, we are transmitting the bacteria, and if they are not vaccinated, it's very likely that they will get the disease and so on. So I will talk about how we will develop bacteria against them, vaccines against them. Next one, please. Okay, go to the next one. In Cuba, here I just want to highlight that there are vaccines against these pathogens in the world, but Cuba does not have them. So Cuba was forced since the 80s to develop the vaccine because we were suffering from outbreaks of meningitis produced by these vaccines, and we were unable, even in the 80s, to produce vaccine. This is the worst situation of the economical embargo, that you cannot buy medicines for your children, and this is the cause that we were forced to develop our own vaccine. Next one, please. I'm just giving the justifications. This is one example to give you a little bit of historical perspective. In the 80s, there was an outbreak of meningitis A and C in Cuba that was controlled. This is the blue curve, was controlled. Cuba was able to purchase a French vaccine, and the outbreak was controlled. But concomitantly, there was an outbreak of meningitis B and C that was unable to control. At that time, there was no available vaccine, and Cuba developed its own vaccine that is called Bamengo, BC, and you can see how the incidence of meningitis significantly dropped after the development of this vaccine. Next one. Okay, and let's go a little bit to the chemistry about this bacteria, Streptococcus pneumonia, Haemophilus influenza, and Neisseria meningitis. These bacteria, they are what we call encapsulated bacteria. So the bacteria is surrounded by a capsular polysaccharide that is what we consider the antigen for the vaccine. So it was very clear and very easy to understand that if you isolate the polysaccharide and you produce a vaccine with that polysaccharide, you can induce an immune property. Take a look to the structure. We as a chemist, we recognize, for example, this is the capsular polysaccharide or Salmonella typhi. Salmonella typhi is the bacteria that produces the typhoid fever and kills millions of children in Africa every year. And that one is the serotype 14 of the pneumococcal bacteria. Take a look that this is a polysaccharide that only contains hydroxyl group. So how can we chemically modify this polysaccharide for producing a vaccine is the topic that I'm going to talk today mostly. The next one, because here I am on the problem of using vaccines only with the capsular polysaccharide is that the capsular polysaccharide, they are what we call T-cell independent antigens. To say in a simple way, they are very bad antigens, okay? And I will explain you why. Next one. There are two different mechanisms, immune mechanisms of how our immune system recognize an antigen. And this is based on the fact that our immune system contains two types of immune cells, what we call the B lymphocytes. The B lymphocytes are the cells that produce antibodies, and the antibodies are what we call the first defense of our body, okay? But then there are a second type of cells that are called the T cells, the T lymphocytes, that they produce what is called the cellular immune response. These are cells which are capable to kill other cells which have been infected. And this is the case, for example, in the COVID-19, it's very important to have both mechanisms of cellular immune response, because if the vaccine, if your antibody do not protect you against the infection, then you will be infected, and then the vaccine has to be able to produce a defense of a cellular defense, okay? And this cellular T-cell response is the one that is able to kill those cells which are already infected. But there is a second mechanism in which the T-cells provide what is called the T-cell help. And this help is a support that the T-cells give to the lymphocytes B, to the factories for producing antibody, which is the B lymphocytes. So take a look that when we use a vaccine only with the capsular polysaccharides, there are no memory B cell response. The antibodies that our body produce are short-lived antibodies. That means that our antibody lasts only for a couple of weeks in our body. We are not protected for the rest of our life, not even for a couple of years. And when we conjugate chemically the capsular polysaccharide to the protein, the immunological mechanisms that is very complex, I am not going to enter into detail. I'm just highlighting what is the need of do a chemical conjugation between the capsular polysaccharide of the bacteria and the protein. Then there is a process that is called affinity maturation. Then there is an induction of a polysaccharide-specific memory B cells. That means that our antibodies, they are not living in our body for years, but we have memory B cells which remain for the rest of our life. And those cells are those that in 10 years, when we are in contact with the pathogen again, are activated immediately, and in a couple of days, they are able to elicit antibodies to defend our body against this disease. That's why memory, what we call the immune memory, is only achieved when we have the capsular polysaccharide from the bacteria chemically conjugated to the protein. And this is the justification of why chemistry is so important in the development of conjugation vaccines, because there has to be a process to chemically conjugate without, and this is very important, without affecting the antigenic properties of the capsular polysaccharide that surround the bacteria, okay? Next one. So this is a process, this is a vaccine that we developed in Cuba from 1985 to 2003, and was an article published in the journal Science. Okay, it's the first only science paper that the Faculty of Chemistry of the University of Havana. I was a student of that group in that year, and this is a vaccine against Haemophilus influenza type B. This is a very, very pathogenic bacteria, but this is a bacteria that has only one serotype. So it's very easy to develop a vaccine against it because it's only one serotype, and does not mutate so much, because you know that bacteria do a lot of mutation. So the chemical process to develop the vaccine is the chemical synthesis of the polysaccharide, and then to do a conjugation chemistry to a protein that is called tetanus toxoid, and this is the protein that is produced in Cuba also for the tetanus vaccine, okay? So it was a very successful vaccine, not only because of the article, but because Cuba was able to produce in 5 years more than 70 million doses to export it to Africa, to export it to Latin America, and to was able to offer to the World Health Organization a more affordable vaccine than the one produced by Pfizer at the time. Okay, next one. This is a highlight in the journal Science, in which they mentioned that the group at the University of Havana was able to develop a vaccine, and they call it a sweet victory for Cuban science. And you know that sugar is very important for Cuba, because Cuba is a large producer of sugar, and that's why they call it a sweet victory for that. They call it without sugar, there is no health in Cuba, or something like that. Next one. Okay, so next one. So this is a scheme highlighting how the incidence of the meningitis and the incidence of pneumonia dropped in Cuba after the introduction of the Neisseria meningitidis vaccine and the Haemophilus influenza vaccine, okay? These are two graphics. Remember that one of them was developed until 2003. But there are a couple of vaccines, which are actually the topic of my talk today, about Streptococcus pneumonia vaccine and the Soberana vaccine. Soberana means sovereign in English, okay, that we developed, and our group contributed to it. We developed against Streptococcus pneumonia and SARS-CoV-2, okay? And this is the topic that I'm going to highlight today. I wanted to give you a little bit of history, how Cuba has a history of 30 years developing vaccines, and we have the conjugation technology and the capacity to produce the bacteria, to fermentate the bacteria, to isolate the capsular polysaccharide, to produce the recombinant proteins to produce the vaccine. But the chemistry has to be very innovative to develop these new vaccines. The next one. So when you take a look, so this is just the vaccines that are in the market. Prevnar from Pfizer, Synflorix from GlaxoSmithKline are international companies which do not sell medicines to Cuba, and also the problem. So these vaccines are considered blockbusters. They sell $6 billion per year, and it's very difficult for an African or a Latin American country to cover all the incidence of these diseases and to vaccinate all the children in 1 year with the prices that these vaccines have. Next one. Go to the next one. So here I just, wait a minute, I just want to highlight what is the challenge of the pneumococcal vaccines. As I told you before, Haemophilus influenza, for example, is a bacteria that has only one serotype in that case. But Streptococcus pneumonia has more than 95 serotypes. Not all of them are equally pathogenic, okay? So you have to decide which are the serotype of a higher incidence in your region or in your country, and then you have to select those serotypes to make what we call a multivalent vaccine. So a multivalent vaccine is a vaccine that contains conjugation, capsular polysaccharide to protein conjugation of each of the serotypes. For example, Prevnar 13 contains 13 different serotypes of the bacteria to protect the children against 13, the most prevalent 13 serotype. In Cuba, the problem when the European or American companies develop their vaccine, they also consider the incidence in Europe or in North America. But the influence in Africa and Latin America and in Asia is different. So we are develop our own vaccines with those serotypes that are higher, they have a higher incidence in our region, okay? Next one. Go to the next one. I want to spend time on this one, okay? Let's go to the, no, next one. Let's go to the chemistry. See, for example, these are the chemical structure of the capsular polysaccharide 7F from pneumococcus, okay? And you see that this is a capsular polysaccharides only containing hydroxyl groups, okay, that are very difficult to modify for a chemical conjugation, okay? So what we do is to do a very well-controlled periodic oxidation. This is a basic reaction that you can do into a molecule. This is a molecule of 100 kilodalton, okay? So this is a molecule that is a macromolecule, macrobiomolecule, okay, that later we are conjugated to the tetanus toxoid. Tetanus toxoid is a protein of 150 kilodalton. This is 150,000 daltons. Most of the molecules that we work in the lab are small heterocycles that they have a molecular weight less than 1,000 dalton, okay? 500, 700, something like that. So you can imagine what is the chemistry, how we implement the chemistry that has to be done between a protein 150,000 dalton with a capsular polysaccharide of 100, from 30 to 100 kilodalton, okay? So we have to use a chemistry that is very efficient, very reliable, and you can scale up. You cannot use a fancy chemistry that only works when you are doing a reaction with milligrams, because then you are in trouble, okay? So the typical process that all the companies use is a typical reductive amination that we know how it works. You preform the Schiff base, the imine, and then you do the reductive amination with sodium borohydride, and then you have a chemical conjugation between the lysine side chains of the amino acids of the protein with the capsular polysaccharide. So you are obtaining a covalent conjugate between the protein and the capsular polysaccharide, okay? This is the state-of-the-art technology of the conjugate vaccine production in the world, okay? Let me show you what we are doing. Next one. So, go back. So, what is the challenge here? The challenge is that to produce a multivalent vaccine, okay, you have to do, for example, if you want to do a vaccine with 10 different valencies, you have to do 10 chemical conjugation process, 10 purification steps, and then you have to formulate one vaccine that contains 10 different glycoconjugates in only one bulk of vaccine. So this is technologically very tedious and very expensive. And that's why the solution that we offer for this and that we patented and that we are introducing into the vaccine development, the next one, is the use of multi-component reaction, and this is one of the biological application of multi-component reaction that perhaps you have not seen before, okay? Take a look to, this is a very simple UGI 4-component reaction. This is a condensation because it produces only water as the byproduct between a carboxylic acid, a primary amine, a carbonyl component, and an isocyanide. The isocyanide, those of you who have worked with that, you know that is a very bad smelling chemical component, okay? So you have to be, but we try to use pegylated isocyanide, lipidated. We have to use high molecular weight isocyanides to avoid the smell of that, because also we are using this chemistry in peptides and protein, okay? I have to say that we are one of the few groups in the world that is able to adapt this technology, the multi-component reaction technology, to the work to the modification of peptides and protein. And this is one example in which we take a normal peptide, we have the lysine side chain with the primary amino group, and we are able to modify the peptide with 2 polyethylene glycol chains. In the talk of Wednesday, I will introduce a little bit for peptide pharmaceuticals, how important is the pegylation process. So it's very frequent that you have to pegylate peptides to make it more metabolically stable, okay? So this is one case in which we produce branch polyethylene glycol into peptides instead of the linear ones, but using the complexity generating capacity of multi-component reaction. We just have to modify the polyethylene glycol with a carboxylic acid, with an isocyanide, and using formaldehyde as the oxo component, okay? This is a reaction that takes place in methanol. It's very simple, but I wanted to show you how we can modify peptides on the side chain. You can imagine that we can do, and we have done the same with protein. So we have patents of pegylated, recombinant pegylated proteins, which have been modified using the multi-component reaction approach. The next one. But also, we can, for example, introduce a lipid and biotin. Biotin is a biological molecule highly relevant. It's very used, for example, for biologists for affinity purification, because biotin has a strong affinity with the streptavidin, the protein that is used for a lot of biological purposes. So we can take a peptide and introduce in only one synthetic step, I mean a lipid and a biotin in only one synthetic operation. Typically, this is done by peptide coupling, and then you have to do one coupling, deprotection, a second coupling, and here we are multifunctionalizing a peptide with two different functional groups using one multi-component reaction. The next one. We have done the same with steroids, with fluorescent label, so we are able to conjugate, for example, a cholesterol and a fluorescent label to a bioactive peptide to see by microscopy where is going the peptide, if the peptide is going to be active on the membrane or in the mitochondria, or if it is internalizing into the cells, okay? And you can imagine the next one that we can use this also for the production of vaccine, because when you take a look to the structure of capsular polysaccharide, as I told you before, there is a state-of-the-art process to introduce carbonyl groups into the capsular polysaccharide, but we also developed a very mild approach in which we could oxidize with tempo the primary hydroxyl groups of the capsular polysaccharide of the bacteria to introduce carboxylic acid components, okay? And there are also some serotypes, like the serotype 9b, that contain naturally the carboxylic acid components, because remember, we need an isocyanide, a carboxylic acid, an oxo component, and an amino component that is provided by the lysine side chains of the protein or the peptide to do a chemical conjugation, okay? Next one. So originally in 2005 years ago, before the COVID, we introduced this method in which we functionalize the protein with a pseudo group, with hydrazide groups, which are more nucleophilic than the normally primary amines. You know why? Because of the alpha effects that makes this amino group more nucleophilic than the typically primary amine. Using acetone as oxo component, we were able to produce conjugates of a variety of capsular polysaccharides to the protein. So we were able originally to demonstrate that our process was equivalent to the process of reductive amination, okay? And that was very relevant because we carried out all the immunogenicity evaluation of the compounds, how once we produce the chemical conjugates, we have to prove that they are equally immunogenic to those that are commercially available by the international companies. The next one. But the most relevant here, so this is the immunological evaluation. I don't want to bother you, but you have to believe me that our conjugates were as immunogenic as look out to the publication if you want more details, because we have enough from immunology for today. Next one. So at some point, what we decided was to use the multi-component power of the UGI 4-component reaction to produce glycoconjugates which enhance immunogenicity. And lipidation is very important for that. So one of the technology that we have very well established in our laboratory is the protein and the peptide lipidation. I will talk a little bit about that about Wednesday, but this is one example in which we conjugate a capsular polysaccharide to a protein and to two different lipids. These lipids, I'm not going to enter into detail, are able to activate a secondary mechanisms of immunopotentiation that enhance the immune response of our body to the glycoconjugates. And this is, I have to say, this is very simple to show on the slide. So this is a protein with a capsular polysaccharide, but these are huge molecules, so we are able to do that in huge molecules, and I am showing here just a simple representation, but as you know, these proteins, they have a lot of lysines, so we are able to modify the protein and to decorate the protein with several copies of the capsular polysaccharide that are shown to the immune system. The next one. Okay, so what is the problem? The problem with the carrier is that, as I told you before, if we want to produce a decavalent vaccine, we have to make 10 different protein conjugations and the purification steps. Our proposal in our patents was to use a multi-component reaction to conjugate two different capsular polysaccharides of two different bacteria to one carrier protein. In that approach, we reduce to the half the number of conjugation steps, and therefore we can produce multivalent glycoconjugates, just like our immune system loves. These are glycoconjugates that have conjugated two different capsular polysaccharides from two different bacterial serotypes to one protein conjugate. So we are able to prepare, for example, a decavalent vaccine using only 5 conjugation steps, 5 purification steps. So we are able to reduce and to simplify the production process of this using the power of multi-component reaction. And this is the first, and I have to say, only example in the world of how multi-component reaction have been used for vaccine development. Next one. So this is the monogenicity in which, again, you have to believe me that we compare the multivalent glycoconjugates with those produced by the pharmaceutical companies. So we have an equal and no significantly different immune response with our glycoconjugates, that they are multivalent. As I mentioned before, they present two different capsular polysaccharides conjugated to the protein, to the immune system. So we reduce to the half the number of conjugation process. We take poorly monogenic glycoconjugates, which could be adjuvated. That means that we are immunopotentiating the fur, and we can cover a vaccine with more bacterial serotype using a reduced number of synthetic operation. And this is the actual advantage of using the multi-component reaction chemistry for the development of conjugate vaccines against Streptococcus pneumoniae. Okay, next one. So we are now developing not only pneumococcal vaccine, but we are developing vaccines against different bacteria. So we want to combine in only one vaccine glycoconjugates from Streptococcus pneumoniae and Neisseria meningitidis, for example, to make a vaccine that is able to protect our children not only against one bacteria, but against two different bacteria. And this will be actually a drastic advance in the vaccine development, because it will be the first time that we can, in one vaccine, not just adding different glycoconjugates, but producing multivalent glycoconjugates that are able to produce not against two different serotypes, but against two different bacteria, okay? Take a look that this looks very complex, but at the end it's the same simple UGI 4-component reaction in which you take a capsular polysaccharide with a carboxylic acid from Neisseria meningitidis, from serotype B. You take also another one with the carbonyl components, and then you use the protein with the amino groups from the hydrazide or from the lysate side chains. The next one. Okay, so we are producing multivalent vaccines against different. At that time, remember that this was obtained, this was patented in 2018. We are advancing the vaccine now to the production technology, but a couple of years later, the COVID-19 appeared in our life, okay? So this is a representation of SARS-CoV-2. As I told you before, the viral infection is mediated by the internalization of the virus inside our cells. So when we want to produce a vaccine, we have to look for B-cell response, which is called the antibody response, but also T-cell response, okay? I don't want to enter into details, but you have to realize that both type of immune response are important. Next one. And that the key for the vaccine production is to stop the interaction between the spike protein. So this is a representation of the spike protein. It's a trimeric protein that surrounds the membrane of the virus. We have to remember that the virus is not a living organism, so it's not a cellular organism. The virus is just a mixture of biomolecules, lipids, oligonucleotides, I mean nucleic acids and proteins, okay? But the spike protein, which gives the name of coronavirus, surrounds the protein. So any vaccine developed in the world, what it's intended to do is to stop the fusion of the virus with with the membrane of our cells, and to avoid the internalization of our cells, okay? So all the chemistry and all the biology that was done was to stop this interaction between the ACE2 receptor and the spike protein from the cells, okay? For that, next one, please. For that, this is the structure of the protein, and this is a lot of protein chemistry that perhaps you are not familiar to it, but you have to know that there was very soon in 2020 recognized that you don't need the entire spike protein for that. So say what you need is what is called the receptor binding domain, that is only a tiny part of the protein, and that tiny part of the protein is the one that interacts with the AC2 receptor, okay? So our approach in Cuba was not to express the entire protein like it was done. I think 90% of the world focused on expressing the spike protein, or for example, the Pfizer and Moderna vaccines, they focus on the RNA, the RNA sequence of the entire spike protein. Our focus was to produce this small protein, the receptor binding domain, okay, and to use it as a vaccine, okay, because we knew from an article that was published very soon that this was the part of the protein. You don't need the entire protein, but you only need the part that interacts with the cellular receptor, and if you block that interaction, then you are stopping the infection from the virus into our bodies, okay? That was the principle of the vaccine development. Next one. Okay, so I don't want to enter into here, but this is a protein that unfortunately is a small protein. It's only 30 kilodalton. You have to know that for vaccines, our immune system, what we like are very large macromolecules, something that actually mimics what we see, what our immune system sees in nature. So small proteins are very poorly immunogenic, okay? So we have to try to make that small molecule more immunogenic. And for that, we use our chemical proposals, okay? The solution, one of the solutions was to conjugate the receptor binding domain protein to a carrier protein or to a lipid adjuvant. Before, I show you how we develop different type of conjugation chemistry to conjugate polysaccharides to protein. In this case, what we have to conjugate was a protein to another protein, but what we wanted to do was to conjugate several copies of the receptor binding domain to the carrier protein that was the tetanus toxoid to produce a macromolecule, a huge macromolecule, which was able to mimic by size and by the presentation the size of the virus, okay? Remember that the virus have several copies of the spike protein surrounding the virus membrane. So we had to be able to build something of that size and in a multivalent fashion, and in a way that we were able to present, okay? Next one, please. So we use a lot of chemistry. This is a chemistry in which we selectively modify the N-terminus of the protein, because the part of the protein that we are highlighting in red is the part that interacts with the AC2 receptor, okay? So if we are going to functionalize the protein to make a protein conjugate, you cannot do it. You cannot do it by the part that interacts with the cellular receptor, because you are blocking the epitopes of the protein that need to generate the antibodies, to make functional antibodies, okay? So the red part of the protein was not possible to be touched during the chemical conjugation. And this is the part of the chemistry in which we enter into the field of site-selective protein conjugation, okay? Which we are able to modify only one residue of the protein without affecting the complete protein structure, okay? So this was a chemistry that was developed by Matthew Francis in the United States. We adapted the pyridine carbaldehyde chemistry that interacts with the N-terminal residue of the protein. Next one. And then we introduce a thiol. Come back, please. We introduce a thiol and acids into the N-terminus of the protein. The next one. Okay, so we also introduce an acid into the N-terminus of the protein using other type of chemistry, and we use the typical bioorthogonal chemistry that received the Nobel Prize 2 or 3 years ago, that was developed by Caroline Bertosset in California in the United States, to conjugate the tetanus toxoid to the carrier protein. The problem was that the linker was immunogenic. That means that the linker, the hydrophobic linker that we use, elicit an immune response per se. So therefore, we don't need antibodies against the linker. We need antibodies only against the protein that we are conjugated to the tetanus toxoid. So we have to discard this strategy and to use the next one please, and to use the next one in which we express a protein containing an additional cysteine that was very far away from the red part of the protein that, remember, is the receptor binding motif that the one does, and we develop a chemistry to develop this intermolecular disulfide bridge without affecting the three-dimensional structure of the protein. It was very easy to tell, but it took us a year in our laboratory to develop this chemistry in which we selectively reduce the disulfide bond in a protein without affecting the other part of the protein, okay? This is the antigenicity evaluation in which we prove that we did not affect the three-dimensional structure and the antigenic properties of the protein. So the protein was still recognized by the antibodies and was still able to generate a antibody response, a functional and neutralizing antibody response. The next one. We published this chemistry in 2021. Next one, in ACS Central Science. Later, with that thiol, we did a Michael addition to incorporate an isocyanide group into the protein. This is the third report in the world describing the synthesis of isocyanoproteins. As I told you before, we typically work with terbutyl, cyclohexyl, or aliphatic isocyanides, but this is one example in which this chemistry also works with proteins. And here what we used was a different multi-component reaction that is called the Passerini three-component reaction, in which this is one example in which this multi-component reaction is capable to introduce two lipidic tails into a protein in only one synthetic operation. And these two lipidic tails, if you remember, we use it also in the UGI reaction because these two lipidic tails are able to activate one specific immune mechanism that they are called self-adjuvation mechanisms, in which what you are doing is you are immunopotentiating your antigen. So you are making your antigen more immunogenic just by introducing two lipid tails. And we were able to do that in only one synthetic operation using the Passerini three-component reaction. This is a multi-component reaction that is like the small sister of the UGI four-component reaction, does not include the primary amino group, but it's just an isocyanide, a carboxylic acid, and an also component. And we can use aliphatic aldehydes and fatty acids, normal fatty acids like palmitic, stearic acids, to be incorporated into the protein and to make it more immunogenic. The next one. This was a second approach of what we use. So this is a immunogenicity evaluation of the conjugate. We call this vaccine Soberana 01 because it was the first candidate that we developed in the Finland Institute of Vaccine in cooperation with our group, okay? And this is the immunogenicity. I don't want to bother you, but you have to believe me that we have a very potent immunogenicity, and this was a vaccine that advanced to phase clinical trials 1 and 2, and was stopped there. And the reason that was stopped was that because we were able to develop a better candidate that actually advanced to phase 3 clinical trials. The next one. But this is a clear example on how multi-component reaction that we typically see it only for heterocycle chemistry, peptidomimetic chemistry, was used for protein chemistry for the production of vaccines as well, okay? So the final protein conjugate that we developed was the modification of the tetanus toxoid with maleimide groups, which is a very powerful Michael acceptor, and then to do the Michael type chemistry to conjugate several copies of the receptor binding domain into a protein, very large protein like the tetanus toxoid. That was a technology that we already knew from before, from the development of the antibacterial vaccines, okay? So we developed conjugation with 2 copies of tetanus toxoid, with 6 copies, with 10 copies, and with 15 copies, okay? We actually varied the range of receptor binding domain that were surrounding the tetanus toxoid. And we found that by addition of only 6 copies of the receptor binding domain, we were able to mimic the nature of the SARS-CoV-2. We were able to present to our immune cells, our immune system, something that mimics the actual virus and is a protein surrounded by the receptor binding domain, okay? This is a very easy chemistry to say, but we have to selectively modify the protein at the positions that we want. So I have to say that it's very easy to sell after 2-3 years of the, but it took us several months for the development of this. So we also proved that our immune conjugates were recognized by the AC2 cell receptors and so on. The next one. So these are the summary of all the protein, all the SARS-CoV-2 vaccine candidates that we developed. So we developed a receptor binding domain conjugated to a carrier protein. We developed what we call self-assembly RBD nanoparticles, receptor binding domain nanoparticle, because this lipidated self-assembly into a huge nanoparticle because they are amphiphilic. They are amphiphilic molecules with a protein in the head and the two lipidic tails that make like a surfactant, like mimicking a surfactant, so they self-assembly into a vesicle-like particle. And we also use some genetic function that is more molecular biology, and I don't want to talk about because this was not developed by our group, okay? So the perspective of the Cuban strategy to develop the vaccine can be found in this article, the chemical part. The next one, please. Okay, go to the next one because this is all the immunological evaluation. And at this point, so take a look to the date of this article. This is an article, this is an editorial article in Nature, because I have to say when we declared in Cuba that we were not buying vaccines abroad, that we were able to produce our own vaccines, we were told that we were crazy. Are you really crazy? Are you able to do it? So this is a question from Nature. Can Cuba beat COVID with the homegrown vaccines? Okay, but also the Lancet, the section in infection disease, Lancet is one of the most prestigious medical journals in the world, also make an editorial article about the successful pandemic response of Cuba, and behind that successful response was our capacity to produce our own vaccines using chemistry, a simple multi-component chemistry, a simple protein modification chemistry, but was a chemistry that we were able to develop over the last 10, 15 years. I call it a simple because now we dominate it, but you have to know that you need a lot of years of development to be able to dominate the protein modification and carbohydrate modification technologies and so on. The next one, and this is the last, but I don't want to bother you, the Cuban vaccines were very successful. Within 6 years, we were able to vaccinate 92% of our people. Go to the next one. So these are the articles, the medical articles were the mostly in Lancet type journal, but also in the Journal of American Medical Association, and so on, with the immune response. Go to the next one because this is too much immunology. I just want to finalize with this slide. So Cuba was without vaccinating anybody, in a very bad economic situation, with the country closed, without tourism, without anything. We closed the airports and everything. And because we were able to produce our vaccines, we jumped into the first place of the vaccination in Latin America, and one of the best also of the first in the world. Our vaccine showed a 92.6 clinical efficiency against the disease. That was very important. That's why I highlight at the beginning against the infection or against the disease. So what you are actually able to mean is that you don't get the disease at this point. So it has a 75 efficacy against the infection and 100% against the death. So after we vaccinate everybody, I have to say that Cuban people did not die from COVID in 1 year. Later the situation changed because the virus mutated a lot. Now the virus that is circulating now is not the original SARS-CoV-2. It's one subvariant of the Omicron that we know that the original vaccines were not developed. So the receptor binding domain has a different mutations, and therefore the antibody response is not efficient enough to protect ourselves against that. Also, it was the first vaccine that was approved for children of 2 years, and the reason for that because the antibacterial vaccines that we use, the conjugation technology that we use was the same using the tetanus toxoid, because the protein tetanus toxoid is used in children, and our children are capable to, so it's a safe protein that elicits a very high potent and functional antibody response, okay? So these are what we call the medical results of our chemical approach to produce vaccines. Next one. With this, I want to finish. I want to thank the University of Havana, the Finlay Institute of Vaccine, that was the actual developer of the vaccine. Our group, our academic group, what we contribute was with the science behind that, but all the factories, the protein production capacities, and so on are in a biotech institute in Cuba. Our group just gave the ideas and the science behind the protein modification and the carbohydrate modification chemistry. Also to our colleagues in Germany, because part of our goal was to be able to compare the efficacy of our vaccines with those of messenger RNA vaccines that were being developed in Germany at the time. So we were able to prove, and you can see it in the articles, that our protein conjugate vaccines were as efficacious as the messenger RNA vaccines. And I want to thank them, also to the Martin Luther University and to the Alexander von Humboldt Foundation, who have funded most of my career and my cooperation with Germany. Thank you very much again for the invitation. I know that today was a little bit too biology. I have to accept it, but I wanted to highlight a little bit how a simple multi-component reaction approach, something that we see only for a small molecule, it can be also applied to very large proteins and the medical application that we are seeking for those reactions on Wednesday. And on Thursday, I will talk a little bit more about pure chemistry, peptide modifications, peptide drug conjugates, and so on, so on. Always with a biological prospect, because our group also developed chemistry looking for good medicines and therapeutics. Thank you very much for your kind attention.
Thank you, Daniel. I think we have some questions from the audience. Please, any questions?
Thank you for your very interesting lecture. It seems like there is a lot of chemistry to apply for vaccine, to vaccine preparations. It's really surprising for me, because I thought that there are just some enzymatic reactions, sort of, but
Everybody
not pure
so.
chemical reactions.
It's only molecular biology and all this stuff. It's not.
Molecular biology and all the methods. It's very interesting. And I don't know, is it an ethical question? But have you tried your vaccine before original trials, medical trials on yourself?
Well, I have to say that the vaccine development have a series of protocols that are very strict, and you cannot violate. So even if people are dying, you cannot speed up the vaccine development.
Yeah, it's prohibited to start.
It's prohibited and it's regulated by the World Health Organization, and you simply cannot do it. So you have to run phase 1 clinical trials, phase 2, 3 clinical trials, and phase 3. Each of them provides a different answer. The phase 1 is with 40 individuals. I participated in the one that I developed, because in Cuba we say that we, the scientists, has to be the first one to be volunteers for those vaccines, because if you don't believe in your product, nobody will believe in that. So it's very common, not only in Cuba, but in many places, that you offer as volunteer for you have to be an adult, healthy individual, and so on, and you participate. And this is only about security. The first one, you only show, because a vaccine has to be safe. It has to be efficacious, but it has to be safe. And the phase 1 clinical trials is only about safety. You have to prove that there are only mild secondary effects. You get some pains on the arm. Okay. And I have to say that the protein-based vaccines are used for children just because of safety. So it would be very, so you see that the Pfizer and the Moderna vaccine were not approved in children after 2 years of being used in adults. But typically that takes 20 years. Typically you take like 2 decades to really prove that there are no secondary effects, because there are some secondary effects that appears after 5 years of using the vaccine, like an autoimmune disorder or something like that. In the case of COVID, people were dying, and you have to accelerate the process. But typically, you have to prove that this is safe in adults, that is efficacy, and then, only then, you go to children. So the reason that why we were able to jump into the children. Our vaccine was the first in the world to be used in the children is because we use the protein conjugation technology that is used in children since 50 years ago. So it has already proven that it's safe. But our vaccines, like all the vaccines in the world, have to run to a phase 2 clinical trial in which you vaccinate 1,000, 2,000 individuals, and then you show the efficacy. You take the blood from the people, you analyze the antibody response, and then you see how functional is the antibody response. Then you present your antibodies to the virus, and you see if the antibodies are able to block the interaction of the virus with our cells. This is called a neutralizing assay, in which you prove that your antibodies from your body after 1 month after vaccination are functional and neutralizing. And then you go to a phase 3, in which you vaccinate 500,000 people. Okay? And that takes, and this is in phase 3 is where you prove that the clinical efficacy of vaccine that is 90, 95, 92. So the Cuban vaccines went through all the process that the Americans, the European, the Russian ones, it has to be done. Otherwise, you cannot do it. And there are regulatory agencies in every country. For example, in Latin America, only 4 countries have medical regulatory agencies: Argentina, Mexico, Colombia, and Cuba. The rest one do not have it because they are not powerful enough. They don't have the regulations and the medical sector with hospitals who runs the clinical trials. It is very strict. I have to say that I was always angry. Why don't you speed up? Don't you see that the people are dying? The doctor said, no. It has to be as it has to be. Otherwise, we have to wait 6 months. Otherwise, we cannot vaccinate people with that.
Okay, I see there are strict rules which shouldn't be violated. And actually, I have another 2 questions.
Yes.
Yeah. Where do you get those polysaccharides which you conjugate to the protein?
Yeah, it's a very good question. So the paper in Science that we published in 2004, we did the polysaccharide by chemical synthesis. This was a very powerful carbohydrate chemistry group that was in Cuba, and the director of that group is Vicente Pérez, is the big vaccine developer in Cuba. But for the Streptococcus pneumonia vaccine, we grow, this is done by microbiologists, we grow the bacteria in a very secure facilities, and then we isolated the bacteria, we kill them with liquid nitrogen with a different process, and then you isolate and purify the capsular polysaccharide. So you are isolating the antigen from the bacteria. And then we do the chemical modification of the polysaccharide, introducing carbonyl components, carboxylic acids, and so on. And we have to do it in a way that we are not affecting the antigenicity of the, so if you affect the structure of the polysaccharides, then the antibody response that you generate is not functional because it's not blocking the colonization of these cells into your body. The antibody has to block and to bind the bacteria and to not let it colonize your body. That's why we have to do, the chemical process has to be very mild. We do a periodic oxidation, but we have to do, this is a lot of stereochemistry, with not the stereochemistry, but stoichiochemistry, in which you have to generate only a minimum amount of carbonyl groups per polysaccharide units, just to, at the conjugation sites, you cannot open all the sugar units because otherwise you are killing the antigenic properties of the antigen. And then the antigen is not antigenic anymore.
Okay, I see. Thank you. And the last question about your toxoid protein.
Yes.
Are there any other proteins which are suitable for conjugation?
Yes, they are. So is also the diphtheria toxoid. Remember that a toxoid, well, probably you don't know, a toxoid is the toxin from the bacteria that is detoxified. So it's treated with formaldehyde and other chemical agents, is heated, so you destroy the functionality of the protein, but the protein keeps what we call the epitopes. The epitopes are the peptide sequence that are recognized by our immune system to elicit an antibody response that is functional, the same that with the protein structure that with the polysaccharide. Okay, so you have to treat chemically the protein to kill the functionality of the protein, the toxicity of the protein, but you cannot destroy the protein in the 3-dimensional structure in a way that you are affecting the epitopes of the protein. And the epitopes are peptide sequence that elicit the immune response against those peptide sequences, and that has to be maintained after the chemical treatment, okay? So you also grow the toxoid bacteria, and you isolate the protein, and you purify the protein by the typical methods. That's why you need a very strong biotech industry that is able to do, because this is biology and microbiology in principle. Our group received the protein already and worked with that, and it's not toxic anymore, it's already detoxified, okay?
Okay, thank you very much.
No, my pleasure.
Any more questions? Just a small one from me, okay?
Yes.
Just a practical one. When you work with biopolymers and you end up with very large molecules, it's hard to analyze it, to understand that you got what you wanted. How do you analyze your conjugates?
It is, and that's why we need a lot of international cooperation, because in Cuba, what we are lacking most is the modern instruments, because this Emaldi instrument to evaluate the molecular mass of the conjugate, it costs 1 million. We don't have in Cuba. That's why we have a lot of international cooperation. You have to realize that the analytics is different. When you work with the small molecules, heterocycles, peptide of small size, use a typical column chromatography with silica, what everybody, I did my PhD with that. Okay, later you work with the peptide of 24 amino acids, with all the lysine and the carboxylic acid side chain deprotected. You cannot put that peptide into a normal silica gel. Then you use reverse phase HPLC analytics, which is also a standard. Sometimes you don't use it but for those who use it. And then we are running with macromolecular conjugates, okay? You conjugate a protein of 30 kilodalton to a protein 150 kilodaltons, and then you purify the conjugate by a chromatographer that is called size exclusion chromatography, that we also received that in is that you purify polymers. And therefore, size exclusion chromatography isolate molecules by size. And then you are able to isolate your conjugate of 450 kilodalton, for example, and to separate it from the remaining tetanus toxoid that did not react, for example, and from the small 30 kilodalton protein which did not react. And this is the way it's done. It's a typical HPLC in the same way, but the principle is just different. The analytics is very different for macromolecules than for small molecules, and peptides are in the middle, okay? That was a very good question, because the students sometimes say, how do you purify those? It's a standard. Sometimes we are not used to it, only that.
Yes. We are not used to that.
Yeah,
surely.
But there has to be polymer groups here who work with size exclusion chromatography and something like that, because with polymerists, this is more common. And macromolecules are large polymers with high molecular weights. Yeah.
Thank you very much.
My pleasure.
So, if we do not have any more questions, I think we can conclude today's lecture saying thank you once again. It was very interesting, very informative. Thank you.